Assalamu alaikum grade 12 students. Today we have to explain documents 4 of chapter 3 detection of a genetic polymorphism. By the end of this video you will be able to define restriction enzyme, recognize that each restriction enzyme has a specific cleavage site, then determine the number and the size of DNA sequences after usage of restriction enzyme, also explain the use of gel electrophoresis and recognize the use of RFLP. In the previous lessons, you have learned that a gene is a specific sequence of DNA that codes for the synthesis of a polypeptide. The synthesized protein is involved in a certain process that affects a certain phenotype of an organism. For example, the purple color of this flower is determined by the protein that makes the pigments in this flower. This protein is coded by a specific gene in the DNA of this flower. You have also learned that a mutation changes the sequence of DNA nucleotides of a gene, and thus change the synthesized protein and thus affects the phenotype, like the white flower in this case. In this example about flowers, and based on what you learned in chapter 2, you can easily know whether a purple flower has only the normal purple allele or it carries as well the mutant white allele in its genotype. Right? Simply make a test cross, analyze the results, and find out the genotype of the purple flowers. But what if the gene under study is a human gene? What if we want to know if this boy with brown eyes carries the mutant allele for blue color? Or what if in a family with a history for a genetic disorder, we want to know whether the fetus has the mutant allele or not? Of course, no test cross can be made in case of a human. Actually, the experimental studies performed on other organisms like plants or animals cannot be applied on humans. Then, how is the genetic variation detected in humans? How to know whether someone has a certain allele or not? The answer is DNA analysis using restriction enzymes. But before we study DNA analysis in human and for what purpose it is made and what techniques does it use, let's find out what are restriction enzymes and how do these enzymes help in detecting the different alleles of a human gene. Restriction enzymes are useful tools used in the genetics lab to study human genes. Those enzymes were first isolated from bacteria. Although they are unicellular, bacteria are subjected to viral infections. Viruses attack bacteria by injecting their DNA into the bacterial DNA. But bacteria can defend themselves against viruses using enzymes that can cleave or cut the viral DNA. By cutting the viral DNA, those enzymes restrict the action of the virus, and that's why they are called restriction enzymes. You must be asking, but why are we talking about bacterial enzymes? What is so special about these enzymes? Like any enzyme, restriction enzymes are specific. They recognize and cut specific sites of the DNA molecule. The table of document A of your books shows some restriction enzymes isolated from different bacteria. Notice that the enzymes are named on the behalf of the bacteria from which they are taken. For each restriction enzyme in the table, you can see which DNA sequence does this enzyme recognize. This sequence is called recognition site. And how does the enzyme cut through this site? This site is called cleavage site. For example, the restriction enzyme EcoR1 recognizes the following sequence of nucleotides in a DNA molecule. We say that this is the recognition site of EcoR1. Once EcoR1 recognizes such a sequence, it cleaves the DNA between the nucleotides G and A. This is what we call the cleavage site of EcoR1. Like all other recognition enzymes, EcoR1 makes a double-stranded cut in the DNA molecule. It cuts between G and A in the two strands of DNA so that DNA fragments are obtained. These DNA fragments are known as the restriction fragments. The number of restriction fragments obtained after the cleavage by a restriction enzyme depends on how many times the enzyme cut the DNA. In this example, EcoR1 has 2 recognition sites within the given DNA molecule. So, EcoR1 will cut twice through this DNA molecule. 3 restriction fragments are obtained, as you can see. So, in general, you can use this formula every time the number of restriction fragments equal the number of recognition sites plus 1. Let's apply this rule on the example of document B of your books. This document shows the restriction fragments obtained after cutting the same DNA molecule with 2 different restriction enzymes, NOT1 and EcoR1. Can you determine how many restriction sites are there for each enzyme within the given DNA molecule? In case of NOT1, 4 restriction fragments are obtained. This indicates that the recognition site of NOT1 is repeated 3 times in this DNA molecule. While in case of EcoR1, 7 restriction fragments are obtained. This indicates that the recognition site of EcoR1 is repeated 6 times in this molecule. So, as you can see, each restriction enzyme acts in a specific way even on the same DNA molecule. So, up to now, we said that restriction enzymes originate from bacteria and they cleave the DNA at specific recognition sites. When an enzyme has more recognition sites within a DNA molecule, then the more it will cut through this DNA molecule. But you still don't get how these restriction enzymes help us detect the presence of a certain allele. Let's say a mutated one, for example. Let's consider this example. Given this normal allele of a certain gene to be treated by the restriction enzyme ch3, the recognition site of this enzyme has the sequence CCGG. How many times will this enzyme cut this allele? How many restriction fragments will be obtained? You must be saying that 3 restriction fragments will be obtained since there are 2 recognition sites for this enzyme within this normal allele. Now, let's consider the mutant allele of the same gene. Can you identify the site of a mutation and its type? Yes, exactly. You can see that a point mutation by substitution has occurred at this level. What if this mutant allele is treated with the restriction enzyme HAE3 now? You must have noticed that because of the mutation in this gene, only 1 recognition site is left for the enzyme HAE3, right? The other is no longer considered to be a recognition site because the sequence of nucleotides has been changed after mutation. Consequently, the enzyme will cut this mutant allele only once, and thus 2 restriction fragments will be obtained. So, actually, using HAE3 to cut this gene allows to know whether the allele is mutant or normal. So, if a person has 2 normal alleles for this gene, then when this gene is treated with the enzyme HAE3, we obtain 3 restriction fragments for each allele and by the result 6 restriction fragments for both. But if a person has 2 mutant alleles for this gene, then when this gene is treated with the enzyme HAE3, 4 restriction fragments will be obtained because this time only 2 fragments for every allele. You see how it was possible to know about the alleles carried by each of these individuals by using a restriction enzyme. Using the restriction enzymes allows to detect the genetic polymorphism. You must be asking now, how can we observe these restriction fragments? This is a very important question since you already know that the DNA molecule cannot be seen with your naked eye. So, smaller fragments cannot be detected with your eyes. Now, if you place a DNA sample in a vial and then add a restriction enzyme to it, you will not notice any difference. Although you know that restriction fragments are obtained in the vial, you cannot see these fragments with your naked eyes. So, using restriction enzymes alone is not enough to identify the presence of a certain allele. That is why the next step in the DNA analysis would be gel electrophoresis. The negatively charged DNA fragments migrates from the negative electrode towards the positive one. The distance covered by the fragments in the gel depends on their size. The larger fragments migrate slower than the smaller ones and thus stay behind. When all the restriction fragments occupy their final positions on the gel, they are still not visible until they are stained with the dye ethidium bromide. This dye binds to DNA fragments and fluoresces under ultraviolet light. At this point, each fragment appears as a band on the gel, and this generated pattern of bands is referred to a restriction map. So, if we go back to the earlier example about the normal and the mutated gene, the restriction map of the normal allele will show 3 bands, while the restriction map of the mutant allele will show only 2 bands. The bands are obtained at different positions due to their different sizes. Gel electrophoresis is not only used to detect the mutations in genes. Actually, there are regions of the DNA that do not code for any protein. These regions are also subjected to mutations. But unlike mutations in coding regions of the DNA, mutations that occur in the non-coding regions do not affect the phenotype at all. However, such mutations increase the genetic diversity such that every individual has his unique DNA that is never identical to anyone else. That's why if the DNA of different individuals is treated with restriction enzymes and allowed to migrate by gel electrophoresis, the restriction maps will show so many differences. Like in this case, the difference in restriction maps obtained called RFLP or restriction fragment length polymorphism. Now, we can say that the genetic polymorphism among individuals is not only detected by phenotypic variations, but can also be observed by RFLP analysis. In the next video, we will see the main domains that use DNA analysis.